In humans, the insulin gene codes for a protein known as preproinsulin, which is 108 amino acids long. The first 24 amino acids are the signal sequence that leads preproinsulin out of the cell. As the protein leaves the cell, the signal sequence is cleaved off, leaving proinsulin, which is stored in the pancreas for further processing. The human DNA sequence for insulin was obtained in mid-80s. Scientists decided to use the human insulin gene and make insulin in yeast. Yeast is a eukaryote. It can do some of the processing necessary to make human insulin. The human insulin sequence was edited so that only the DNA sequence of proinsulin remained. This meant that the first processing step of preproinsulin to proinsulin was skipped. The proinsulin sequence is inserted into a plasmid. The recombinant plasmid is transformed into yeast. Yeast can now make proinsulin. The proinsulin made by yeast is processed much the same way as human proinsulin. Proinsulin folds into a looped structure and disulfide bridges are made between cysteine amino groups. A 33 amino acid stretch is cleaved off, leaving the mature insulin protein. Bacteria, unlike yeast, cannot process proteins. Insulin is made in a different way when using bacteria. Synthetic human insulin was the first golden molecule of the biotech industry and the direct result of recombinant DNA technology. Currently, millions of diabetics worldwide use synthetic insulin to regulate their blood sugar levels. Synthetic insulin is made in both bacteria and yeast. How synthetic insulin is made using bacteria? Watch our video. The link is in description.